It's my great pleasure, excuse me, to welcome our first speaker, Dr. Tom Scammell. Tom Scammell received his Doctor of Medicine, his doctorate degree from the University of Massachusetts Medical School, where he trained in internal medicine. He also completed a residency in neurology at the University of California in San Francisco and a fellowship in sleep medicine at Beth Israel Deaconess Medical Center. Dr. Scammell is a professor in neurology at Harvard Medical School and at Boston Children's Hospital. He serves as an ad hoc reviewer for New England Journal of Medicine, Annals of Neurology, Nature, Cell, and Neuron, and was deputy editor of Sleep. He was a member of the International Classification of Sleep Disorders Task Force, the Sleep Research Society Board of Directors, and the APSS Program Committee. He's authored over 150 journal articles and chapters, and Dr. Scammell has lectured both nationally and internationally on narcolepsy and the neurobiology of sleep and wakefulness. His current research interests including how loss of orexin signaling results in sleepiness and cataplexy, and how orexins affect breathing and development of new medications for narcolepsy. Thank you, Dr. Scammell.
So I'm looking at the title of this talk and realizing that I can talk about the science of narcolepsy, but as you all can imagine, the science of idiopathic hypersomnia is really challenging. But I'll tell you, we have some ideas and we'll get to that. So the overall structure of the talk is to discuss, first off, what causes narcolepsy, and then we'll share some ideas around idiopathic hypersomnia. I'm also going to just touch very lightly on Kleine-Levin syndrome, which is another one of the sleep disorders that's relevant to this. But most importantly, I want to also frame how some of the meds that are used in these disorders work in the brain, because I think when it comes to thinking about science, while we don't necessarily understand what fully causes some of these diseases, we do have a pretty good idea of how some of the meds work. Not all of them, but some of them. It's pretty clear that there's at least two types of narcolepsy. And I think these are now often referred to as NT1 and NT2. NT1, I would say we actually have a very good understanding of. So this is narcolepsy with cataplexy. And people who have narcolepsy type 1 are basically, sleepy every day. And they have cataplexy and a positive multiple sleep latency test. And so, telling your doctor that you've got cataplexy is all fine, but we need objective evidence to help confirm that. And so the multiple sleep latency test is very helpful in that regard. And what we're looking, when we talk about a positive MSLT or multiple sleep latency test, it means that across the 5 nap opportunities, the person falls asleep in less than 8 minutes, and at least 2 of those opportunities contain REM sleep. And newly added in the last few years is if somebody has an overnight polysomnogram, a sleep study the night before, if they go into REM sleep within 15 minutes at night, that's counted towards the daytime tally. As it turns out, going into REM sleep really fast at night is super unusual, and it really only happens in narcolepsy type 1. And so it's a really strong actual observation if it occurs. Another way we can diagnose narcolepsy type 1 is by doing a spinal tap and measuring orexin levels in spinal fluid. The advantage of this is it gives you a really unambiguous answer. If somebody's CS, spinal fluid, orexin level is low, they've got narcolepsy type 1. There's basically nothing else that does it, with some super rare exceptions. The trouble, of course, is it requires a lumbar puncture. And I have to say, a lot of people are hesitant around that. But getting a lumbar puncture is about as uncomfortable as donating blood, maybe a little more so. It takes a couple hours in clinic, and you get a really definitive answer. And so I have to say, in my clinic, we don't do it too often, maybe half a dozen times a year, but it's often helpful when somebody's got a story that doesn't quite fit the mold for typical narcolepsy type 1, and it really just helps really firm things up one way or the other. Narcolepsy type 2 is considerably more mysterious. The definition that doctors use is chronic sleepiness, just like narcolepsy type 1, and a positive multiple sleep latency test. The trouble with this is there's many ways that people can have a falsely positive or falsely negative multiple sleep latency test. If somebody comes into the lab and it's noisy or they're anxious, they may not sleep well. On the other hand, if somebody's taking meds or they stopped meds recently, that can give you a falsely positive MSLT. So we're very dependent on the multiple sleep latency test. And as a consequence, I worry that we lump together a variety of different causes of sleepiness under narcolepsy type 2 that may or may not belong there. And in fact, it's kind of a spectrum. We'll get to the details of this in a little bit. But Narcolepsy type 1, usually clear, unambiguous diagnosis. Narcolepsy type 2, yeah, moderate confidence oftentimes. So, the huge development in the field occurred about 25 years ago when Emmanuel Mignot and Seiji Nishino measured orexin levels in people with narcolepsy in comparison to controls. And what you can see is each dot here represents one spinal fluid sample from different people. And so on the left, you can see in healthy control individuals, orexin levels are typically around 320 picograms per mil. That's a really low concentration. It's really hard to measure, but there's a good test for it now that can be done at Mayo Labs if you wanted to pursue this. People who have narcolepsy with cataplexy, 90% of the time their orexin level is low, meaning less than 110 picograms on this assay. Interestingly, there are some people who have very convincing stories for cataplexy and so forth who have normalish orexin levels, and I don't really know what to make of them. Maybe they just haven't fully lost the orexin neurons, or maybe there's something else going on. But in narcolepsy without cataplexy, that's narcolepsy type 2, 90% of the time the levels are normal, and they are not orexin deficient. You'll see in this example, there's 3 subjects who had low CSF orexin levels. And what's interesting is if you take those people who have a clinical history of sleepiness, but no cataplexy, in which case we'd diagnose them with narcolepsy type 2, if it turns out that they have low orexin levels, and then you follow them for the next 20 years, at least a third of them actually go on to develop a rare event of cataplexy. And so it may be that there are some people with narcolepsy type 2 who just are kind of on the borderline and don't really have a lot of cataplexy, but they are orexin deficient. This loss of orexin in the spinal fluid is loss of the neurons in the brain that make this neurotransmitter orexin. And so on the left, you can see it's called a coronal section. So it's the brain cut this way, kind of like ear to ear. And then we're looking at half of the hypothalamus. So it's basically, imagine this as we're looking at half of the brain in a slice that kind of goes from ear to ear. At the bottom is something called the mammillary body, which is basically a lump that looks like breasts on the bottom of the hypothalamus. And up above the fornix there, you see a lot of green dots. That's a normal cluster of orexin neurons. There's typically about 80,000 of these cells in a normal human brain. But in somebody who has narcolepsy type 1, there's a gigantic loss. 90% loss, 95% loss. We usually see just a few scattered cells sitting in that part of the brain. When this was first discovered by Christelle Peyron and Jerry Siegel, one of the questions that came up is, well, maybe the orexin neurons are still there, but they're just not making orexins anymore. And Mady Tafti had a paper on this a couple of years ago where he makes an argument for why the cells may be there, but they're silent, they're not making orexin. However, we did a study where we looked at other markers of these cells. As it turns out, they make other neurotransmitters. And when we look for those, they're also gone. And so I think anyway, my personal feeling is that I think the cells are gone and they're not coming back. But I may be wrong. I have to say, if the Tafti idea that the cells are just somehow silenced, if that turns out to be true, that would be great, because there may be a way to kind of rouse these cells again, though that's not at all clear how to proceed. So what kills the orexin-producing neurons? So here I'm going to do a little immunology 101 with you guys. Normally, when you get an infection, whether it's a bacteria or a virus, we call these pathogens. And so up in the top of the figure there, you can see these nasty little things, these virus molecules or bacteria. These get eaten up by what are called antigen-presenting cells. You might have heard of them like macrophages. And basically what they do is they gobble up bacteria and viruses, they break them into little pieces in lysosomes. They then take those fragments of the bacteria and they actually push them out onto the surface of the cell, and they're expressed along with what are called MHC class II molecules. These are HLA molecules. If any of you have had HLA testing to look and see if you have the gene that runs with narcolepsy, it's that thing we're talking about, the MHC class II molecule. People with narcolepsy have a very particular gene that is very common in that group. Anyway, so these macrophages, these antigen-presenting cells, gobble up these bacteria. They take little fragments, stick them on the surface, and they basically are waving them out there like a little flag to T cells. And then a T cell comes along, and if the T cell has the right receptor, it binds to that piece of flu or virus or whatever it is on the surface. And it's like, oh, this matches my receptor. The T cell becomes activated. It makes these chemicals called cytokines that trigger an inflammatory response, and those cytokines kill the bacteria. And so you got these macrophages that activate T cells that produce cytokines that kill the bacteria. That's normal. That's good. That's how you fight off infections every day. In narcolepsy, we think that those T cells accidentally attack the orexin neurons as well. And we're not actually sure of the specifics. Emmanuel Munoz done some nice stuff showing that there are some molecules on flu virus that share a lot of similarity with the orexin molecules themselves. And so these cells may think they're fighting off flu, but they're accidentally killing the orexin neurons. And as a consequence, once you have that inflammatory attack against the orexin neurons, those cells are gone. There's different kinds of T cells. And in this case, they found CD4 and CD8 cells that are activated by the precursor for the orexin molecules themselves. That's a little scientifically heavy, but the general theme of this is something we call molecular mimicry, where the T cell is normally responding to this, just for example, virus piece, but then accidentally it cross-reacts with this other thing, a state of mistaken identity. Okay, so now this is a much harder question. So I think at this point, we're pretty comfortable about what causes narcolepsy with cataplexy, but what causes narcolepsy without cataplexy or NT2 is still anybody's guess. And part of the problem is that at this point, I think maybe 20 brains have been described after people have died after a lifetime of narcolepsy type 1, but there's only been a handful for narcolepsy type 2. And so we don't have a good understanding of the neuropathology of like, what's it look like under a microscope? Are the orexins there or not? Is there something else going on? Jerry Siegel had the only paper on this where they described 2 patients with narcolepsy type 2. One of them had a 33% loss of the orexin neurons, which to be honest isn't going to do a whole lot. The other one had 85% loss. And if you measure spinal fluid levels, about 20% of narcolepsy type 2 patients have lowish levels. And 13% are in the intermediate range, where it's kind of a gray zone that's a little hard to interpret. And it does, so one could look at this and actually think, well, maybe this is just all due to partial loss of the orexin neurons. It's not so severe as to cause cataplexy, but it's bad enough to cause sleepiness and other stuff. So, and as I mentioned before, some of those people who have low orexin levels will go on to develop cataplexy in the coming years. So that's one idea. Another idea is that we had talked about for a long time is that, well, maybe people with narcolepsy type 2 are perfectly great at making orexins. They make normal amounts, but the orexin receptors just don't work so good. And the argument against that is that Centessa, Takeda, Alkermes are now doing research with orexin agonists. These are drugs that mimic the effects of orexins. And they work just as well in narcolepsy type 2 as they work in other people who have sleep disorders. And so that really suggests that it's very achievable to activate the orexin receptors in narcolepsy type 2 patients. So I don't think it's a downstream signaling problem. The other possibility is that it's just a disease that looks kind of similar, but it's got nothing to do with orexin. And in that regard, we have no leads to go on. So now we get into the really murky part of this all, which is idiopathic hypersomnia. And I think as many of you appreciate, the name itself sort of is an admission that we have no idea what's going on. Hypersomnia in doctor language means sleepiness, and idiopathic means we have no clue what causes it. And so it's a bit of a broad term that if somebody doesn't really fit into a narcolepsy category, if they don't really fit into something else, if it's clearly not meds, et cetera, we just throw up our hands and say, well, maybe it's IH. And in the most classic form of idiopathic hypersomnia, people have, of course, chronic sleepiness, but they'll also have long sleep periods at night. They can sleep 12 hours at night, and it's really hard to wake up in the morning. They set 4 or 5 alarm clocks. If they take a nap, sleep for 2 or 3 hours, they don't feel good afterwards, which is quite a contrast to narcolepsy, because people with narcolepsy will sleep for 15 minutes and pop awake and feel really good. And so people with idiopathic hypersomnia often have this sleep inertia, where once they fall asleep, they stay asleep, and they just keep going and going. So if we hear that history of really long sleep times and really awful sleep inertia, personally, I feel much more confident that this is probably idiopathic hypersomnia. But sometimes people are just tired, and they don't take long naps. Their nighttime sleep isn't long, and there's a great, huge overlap between idiopathic hypersomnia and narcolepsy type 2, where we just can't really necessarily categorize the diagnosis all that precisely. But as with narcolepsy, we want to hear that history of sleepiness from a patient, but we also need to see some objective evidence. And so here again, we do the multiple sleep latency test, where we measure people's propensity to fall asleep. And again, you need to fall asleep in less than 8 minutes, but there's no there's no REM sleep weirdness in idiopathic hypersomnia. You know, people with narcolepsy can easily go into REM sleep. They do it in their daytime naps. In idiopathic hypersomnia, that just doesn't happen so much. And so you can have REM sleep in one of the 5 daytime naps, but not more than that. Now, honestly, the way I really like to diagnose idiopathic hypersomnia now, we're starting to do it in our lab, is to do long sleep recordings and just let people sleep ad lib. So we bring them into the lab, you know, hook them up 10 o'clock at night, turn out the lights, and just say, go sleep as long as you want. And what we look for is more than 11 hours of sleep over the course of 24 hours. Yves Dauvier has done some nice work showing that this is actually a really good diagnostic way of measuring at least the classic idiopathic hypersomnia phenotype of these super long sleep times. Now, in theory, one could do this with like a Fitbit at home or a sleep log, but because those aren't super accurate and rigorous, we'd rather get the objective data in the lab. But personally, I like this idea a lot because what it does is it's helping us identify a more, you know, let me back up a second. With the multiple sleep latency test, we're looking how quickly can you fall asleep? And if you do fall asleep, does it include REM sleep? Well, that's great for narcolepsy, but that's not really testing what's relevant for IH. You know, if it's a really classic IH story, I want to see that evidence of super long sleep times. And if they do fall asleep later in the afternoon and sleep some more, is it a 2-hour nap or something like that? That's very compelling. I was actually just looking at a sleep study before coming to the meeting of a woman who slept 13 hours. And, you know, she sort of woke up for maybe half an hour in the morning, went back to sleep and just kept charging away. So I think that you can think of narcolepsy as being a situation where it's hard to stay awake. In idiopathic hypersomnia, it's hard to wake up. And I think that's, so even though we sort of call these both hypersomnias of one sort or another, I think just, at least in the extremes, they can be distinguished by these features. So this table summarizes what we've covered on the last few slides. So everybody with NT1, NT2, and IH has excessive daytime sleepiness. That's the doctor term for just feeling sleepy all the time. Only NT1, narcolepsy type 1, has cataplexy. Orexin deficiency is only in narcolepsy type 1. 2 or more sleep onset REM periods is seen in either form of narcolepsy, but not IH. There you can only at most have 1 REM episode during the day. Long sleep is common in IH, but quite rare in NT1 and NT2. And sleep inertia is very common in idiopathic hypersomnia. You see it sometimes in NT2, but it's actually really, in my experience, pretty rare in narcolepsy type 1. Most people with narcolepsy type 1 wake up pretty easily in the morning, but then at 10 a.m., they're like, ugh, I'm ready to go back to bed again, and they want to take a nap. But sometimes the first couple hours of the day are the most productive for them, whereas the opposite would be the case for somebody with IH who finds the first couple hours of the day just useless and just they're just, you know, barely awake. So, all right, this is some objective differences that you can see between these. Where we're looking at distinction between narcolepsy type 1, type 2, and IH. Narcolepsy type 1 is pretty much equal between the sexes. There may be a female predominance in IH, but I'm not sure. Honestly, it's really hard to study this. And I think to some degree, it may be sort of a self-reporting thing. And we tend to see more women coming to clinic with this. And maybe that's real in the sense that that's truly what's happening. Or maybe guys just don't come to clinic for some reason. Mean sleep latencies on the multiple sleep latency test. And so that is over the 5 naps, we measure how quickly do you fall asleep in each nap. On average, it has to be less than 8 minutes. But in narcolepsy type 1, they can fall asleep fast, 2.5 minutes on average. Narcolepsy type 2, still fast, 4 minutes. Idiopathic hypersomnia, it's about 5 or 6 minutes. And so the intensity and rapidity with which people can fall asleep varies between these. At the bottom, you can see insufficient sleep. And I bring this up because, of course, it's the most common cause of chronic sleepiness. You know, if you just went out into the general population and found people who said they were sleepy, most of the time it's just going to be they're getting 6 hours of sleep at night. And in that case, they don't fall asleep quite as rapidly. They don't have the REM sleep on their multiple sleep latency tests, and they show a typical frequency of this gene that runs with narcolepsy. What you can see in the top right there is this gene called HLA DQB1*0602. And this gene is really, really strongly associated with narcolepsy type 1. About 90 to 95% of people with narcolepsy type 1 carry this. If you remember that immunology slide I showed earlier, that molecule 0602 is crucial for those antigen-presenting cells to share those little peptide fragments with the T cells. And so it's probably really really key on that whole mechanism. Narcolepsy type 2, they actually do have that gene more common in the general population, 40 to 50% of the time. So it does kind of fit with the idea of at least some attack on the orexin neurons. But idiopathic hypersomnia and insufficient sleep, the frequency of this gene is the same as the general population, which is about 12 to 25%. Oh, by the way, if you haven't had it done, you can talk to your doctor about it. Most doctors don't test for that gene. I actually find it very helpful, particularly if somebody comes in and says, oh, I have narcolepsy type 1. And we're like, maybe, but there's some aspects of the story that are a little atypical. We'll often test for that gene. And if they don't have it, that would be an argument against narcolepsy type 1. If they do have it, it says, oh, okay, well, that's typical for narcolepsy type 1. And sometimes we'll do it in preparation before doing a spinal tap for measuring orexin levels, because orexin deficiency is seen almost only in people who carry that gene. Okay, so circling back to this is supposed to be about the science of these sleep disorders. And as I said, it's a little bit of a struggle when it comes to idiopathic hypersomnia, but I wanted to share a few of the ideas that are out there in the community. One that comes from the Emory University group is that maybe there's something that enhances GABA signaling in the brain. I think you might be familiar with GABA as a neurotransmitter in the brain, a signaling molecule, and it promotes sleep. And so drugs that enhance GABA signaling, like benzodiazepines, like diazepam, clonazepam, those increase signaling through the GABA A receptor, and they produce, pretty striking sleepiness. And so the Emory group asked, is it possible that people with idiopathic hypersomnia are actually making some kind of chemical that sort of mimics benzodiazepines? In other words, it's enhancing GABA signaling. And so about 15 years ago, they collected spinal fluid from people with idiopathic hypersomnia and similar disorders, and they found that there actually was something, there was some chemical there in the spinal fluid that seemed to do that. And it's a really interesting observation. It's been a little bit of a challenge to kind of make the next steps because they've tested some medicines that reduce GABA signaling to see how helpful they are. And some of you might have even taken these, things like flumazenil and clarithromycin. And what's interesting is they've been able to show that people subjectively feel more alert when they take these medicines, but it's been harder to show objective improvements. And so if you do a multiple sleep latency test, people still fall asleep on the flumazenil and so forth. And so to be honest, I don't think we've heard the end of this story. I think it needs more work, and I'm very appreciative that David Ryan, Lynn Marie Trotty are still working on trying to tie a bow around this and figure out what's truly going on. Now, another idea is that maybe there's some kind of chronic inflammation. Because, of course, when you're sick, if you have the flu, I mean, I'm talking about just the general population. If somebody has the flu, they might go and just sleep 14, 16 hours. And so those chemicals, like the cytokines that are involved in fighting off infections, are also very sedating. And so some of the ones that have been studied in animals include things like tumor necrosis factor alpha, interleukin-1, there's something called interleukin-6. And all of these, if you squirt them into the brain of an animal, they'll go to sleep. And so, in clinic, we often hear from patients with IH about that they have other inflammatory things going on. They have some sort of gut inflammation. Maybe they have gluten intolerance. Maybe they have something evidence for chronic mononucleosis. Maybe they have some joint inflammatory process. And so it wouldn't surprise me if at least some IH patients have persistent activation of the immune system, which is contributing to their feelings of tiredness. The important thing here is these two ideas are completely different. In one case, you want to do something to change GABA signaling. The other, you want to do something to change inflammatory pathways. And so it's clear that I hope in the coming years, researchers will be able to start really separating these out and testing these ideas more rigorously. Now, here's a couple other ideas, which I think are also really worth considering. So in most people, you have a habitual waking time. Like, I just came from Boston yesterday. I woke up at 4 in the morning here. And because that's your internal circadian alarm clock. And but one idea is that maybe that circadian waking signal just isn't working right in idiopathic hypersomnia. And so, even though it's supposedly the habitual waking time, the alarm just doesn't ring internally. And it doesn't, and what we call circadian waking drive doesn't really kick in and help rouse you over the coming hours of the morning. And so there's not a lot of evidence for this, but one of the reasons that this idea comes up is because, another thing that we have to distinguish from IH in clinic is something called circadian phase delay, where somebody's body clock is just running late. Like, for instance, this is super common in college students, right? They can easily stay up till 2 in the morning, but they don't want to get up until 10 or 11 in the morning. And if they have to get up at 7 or 8, it's really hard because their alarm clock is set for later. But I'm not saying that people with IH have circadian phase delay, but I think they could lack that alarm clock signal that helps rouse people normally at the desirable time. So in support of this, some people with IH do have a variation in one of the genes that regulates circadian rhythms. It's called PER3. And when people have taken skin cells from some people with idiopathic hypersomnia, they have shown, so just like your whole body, every cell in your body actually has rhythms. And so if you take some skin cells and grow them in a dish, you can actually see that their activity is rhythmic. And it turns out that instead of having a 24-hour rhythm, the skin cells of some people with IH have a longer rhythm, meaning that instead of the alarm clock going off at 7 in the morning, it's going off at 7:30 or 8 or something like that. Also, there's a little bit of evidence that maybe there's a melatonin problem. And so, for instance, melatonin is kind of a sleep-promoting chemical that's released while you're sleeping at night. It then falls over the course of the night so that when you're waking up in the morning, your melatonin is very low. But there's been a little bit of research in idiopathic hypersomnia that maybe that melatonin doesn't just fall. And so there's still some persistent melatonin tone that trickles over into what should be the waking period. The last thing on here is one that I find almost, honestly, like the biggest challenge in clinic, which is that a lot of people with IH express symptoms of depression, and it's really hard to figure out what's the chicken and the egg, because oftentimes they say, oh, my life is so messed up because I'm sleepy all the time. I don't have enough hours in the day to get things done, and that makes me depressed. But of course, depression can also, at least in some people, contribute to feelings of sleepiness. And so in atypical depression, where people don't have a lot of sadness and tearfulness, they can still have fatigue and tiredness. And so, if you just take the population of people with depression and anxiety, half of them report sleepiness. And when somebody's really depressed, it's hard to get up in the morning. And so this is something we talk about in clinic. Honestly, it's a hard thing to unravel, but sometimes the solution is just like, okay, we don't know if the depression is the cause of your IH, but it seems to be at least an ongoing factor. So let's work on these simultaneously. We'll give you drugs to help wake you up, but also I want you to work with your psychiatrist or psychologist on the depression as well, and going on both paths in parallel is often beneficial. Okay, this is in some ways kind of the coolest of the sleep disorders. It's also horrible in its own right. Kleine-Levin syndrome. I don't know if any of you have talked to people who have this. It is weird. This is not a problem of daily sleepiness. It's a problem of episodes of sleepiness. It's sometimes called the sleeping beauty syndrome. And so what happens is it's most typically occurs in teenagers, and a completely normal teenager will just suddenly start sleeping 18, 20 hours a day, and they'll do that for a week, and then they suddenly get better. And it's crazy because they just can't wake up during that time. It's very deep sleep. The episodes can last a week, sometimes at most 2 weeks. They can have 3 or 4 episodes a year. And in between the episodes, they're fine. In teenage girls, sometimes it's triggered by their periods. And in that case, it actually can get better with birth control pills. So it's suggesting there's some hormonal trigger to it. But honestly, it's the most strange thing that you have a completely normal person who just flips into this super sleepy state. And during it, they're also a little kooky. I'm sorry, that's not a great sound. Doesn't sound very medical, but like their appetite can be voracious. People can just eat tons of food, and they can eat things they wouldn't normally eat. They can be hypersexual. That's usually in the teenage boys, which maybe that's not too surprising. They can be irritable. They can be aggressive. And if you ask them, well, once they come out of these episodes, well, how did you feel during this episode? They said that they felt so apathetic. They felt confused. They felt like they were in a dream for a week. There's a feeling that things just aren't even real. They might be hallucinating. It's a very strange state. And so, the trouble is it's super rare. At the bottom, you can see here it occurs in maybe 2 in a million people. And so almost no doctors have even heard of this. And so families are often really struggling to get a name to this. And the good news is that it usually goes away in time. Usually it'll go on for maybe 4 or 5 years. And so that, in the first year, there may be, say, 3 or 4 episodes. And then the next year, there's 3 episodes, and then there's 2. And it kind of improves over time. But what the heck is causing it is a mystery. Interestingly, remember how we talked about idiopathic hypersomnia maybe being more common in women? Kleine-Levin seems to be a bit more common in men. It's also more common in Ashkenazi Jews, which certainly suggests a genetic connection there. And every once in a while, very uncommon, it can run in families. And so there's a few families who've had a couple kids with this. So as you might imagine, we still don't know what causes this. And as a doctor and researcher, I just want to scream about this because it's like, here you got somebody who's completely normal, and then they're just completely flattened by their episode of KLS. It's like, come on, we can figure this out, measure something. And yet people have tried, and it's really hard to say. On the right, you can see spinal fluid orexin levels, and they're kind of lower during these hypersomnia episodes compared to when they're out of it. But they're not super low. They're barely, one or two subjects, they're down in the narcolepsy range or less than 100. But most of the time, they're kind of in the intermediate range. And so I don't know if this is a cause of the sleepiness or if it's just a consequence where it's like they're so drowsy, their orexin system is just kind of quiet. Proteomic analysis means looking at proteins that are expressed in spinal fluid. And I've listed a bunch of proteins there. What they do is anybody's guess. When they look in blood, they found a few other things. Emmanuel Mignot found a linkage to a gene called TRANK1, and that's interesting. That gene seems to increase the odds of getting Kleine-Levin by about 50%. I mean, that still doesn't mean you're very much at risk, because remember, it's a 1 in a million kind of disease. But what's interesting with TRANK1 is that gene is also linked to bipolar disorder and schizophrenia. And the thing that I think is interesting about bipolar disorder is, people have periods where they're manic, and then they have periods where they're depressed, and they can flip back and forth very quickly. And that sort of state switch, Kleine-Levin kind of sounds like that a little bit, like you're going between these two weird, stable states. But what causes that switching is not at all clear. Okay, importantly, and particularly for any of you who have been diagnosed with idiopathic hypersomnia, it's just really good to think about, is there anything else that we should be considering? And so this is a list of just some of the other stuff that your sleep doc should have been discussing and considering. And as I mentioned earlier, the most common cause of chronic sleepiness is just simply insufficient sleep. The average American adult gets 6.75 hours of sleep on a typical work night. And that's just not enough for most people. Most people need 7.5 or 8 hours of sleep. And so chronically insufficient sleep on weekdays and weekends may be a factor that's relevant. Important to rule out other things that produce poor quality sleep, like obstructive sleep apnea, periodic limb movements, pain, right? One of my sleep doctor colleagues got a rotator cuff injury. Actually, she had it on both sides, and so she couldn't sleep on her side. On either side, it was horrible, and she was really messed up by that. Circadian rhythm disorders, I've touched on a little bit already. But remember, if somebody's body clock is running on the later side, it's hard to wake up. They may not be easily awake at 7 or 8 a.m. They don't really start feeling awake until 10, 11 a.m. And so that can be easily sorted out by doing sleep logs and things like that. Sedating medicines, I've mentioned some of the more common ones here, like benzodiazepines, antipsychotics, epilepsy medicines, but there's others. And sometimes people will be on an antidepressant, which doesn't usually make people sleepy, but maybe in a particular case it could. And so it's really worth thinking about meds and if they could contribute. Neurologic diseases are usually pretty easy to rule out because they're obvious. Parkinson's, Alzheimer's, multiple sclerosis. Usually those are not subtle, and it's pretty obvious if somebody's got that. Traumatic brain injury, it's worth mentioning here because this often comes up in the clinical context where somebody say, oh, I had a head injury and then I'm sleepy afterwards. And that actually is fairly common. About 25–30% of people who have a serious concussion can be sleepy for quite a while afterwards. Prader-Willi syndrome is the most common genetic cause of obesity. Again, if it's not going to be missed. It's something that's apparent in childhood. And then, as I mentioned, this overlap with psychiatric disorders. I talked about depression. I also want to just mention anxiety too, because when somebody's anxious, it's exhausting. Their brain and emotional systems are running at overtime. They're just going so fast. And it's not unusual to hear somebody say, oh, things were so stressful. They come home, they just need to take a nap. It's like, yeah, anxiety does that. And so I think that sometimes when people have generalized anxiety, it can contribute to feelings of chronic fatigue and sleepiness. Schizophrenia, that's not going to get missed. But interestingly, schizophrenic people often do have a lot of sleepiness, and it's not just from their schizophrenia meds. And then I don't test for these routinely, but it's sometimes if we're really stretching and trying to figure things out, we will. We'll check for low thyroid activity. We'll check if somebody's adrenal glands are underactive. We'll check if their kidneys and their liver are working okay. Obesity, interestingly, can also just in itself cause some sleepiness, but not at the degree that somebody's going to have crushing sleepiness, but it does contribute a bit to some fatigue and so forth. Okay, so I want to just wrap up on sort of practical stuff of how we treat the sleepiness and so forth since we're trying to focus on the science. This is a list of the meds that are typically used in clinic for sleepiness. And some of them, like methylphenidate and dextroamphetamine, have been used since the 1930s and 40s. And for narcolepsy, these can be pretty good. They come with a lot of side effects. They're very inexpensive. We never give more than 60 milligrams a day because above that, it's just diminishing returns and people often end up with rebound sleepiness. Personally, I've not seen the greatest responses to amphetamines in idiopathic hypersomnia. It's okay. We usually try it. If somebody does it okay on it, then that's nice. But I think we're more likely to see positive results in narcolepsy than IH. Modafinil and armodafinil are really popular, and they're often first-line drugs. And the reason is because they're pretty effective and have very low side effects, much less trouble generally than you get with something like the amphetamines. 15% of people get headaches, 5% can get nausea, but usually those clear up over time. It's a little expensive, but not too bad. Oxybates are, I would call it sort of the big guns of treating sleepiness. These are medicines that you give at night. It produces super deep sleep. And then by some magic that we don't understand, over the course of weeks and months, people start feeling better during the day. They have less sleepiness, less cataplexy. However, these drugs are really expensive, and they produce the highest incidence of side effects of anything that we've got. And they're so sedating, we honestly worry about them. If you had a few drinks and then took oxybate, we would honestly worry about whether you're going to have so much depression of your breathing you could really get in trouble. I've had 2 patients in 20 years end up in the emergency room from excessive sedation from their oxybate. So when I prescribe modafinil for a patient, it's just a 5-minute conversation. It's very straightforward. When I prescribe an oxybate, it's a 30-minute conversation because there's so many issues about dosing it properly and taking it safely that need to be covered. Solriamfetol and pitolisant are new meds in the last 3 or 4 years, and they're both pretty good. Solriamfetol is given at 75 or 150 milligrams each day. It's got pretty good potency and relatively low side effects. It's also medium expensive. Pitolisant is also a nicely tolerated medicine. It's got a completely different mechanism of action from the other medicines, and some people love it. Nice medicine, can be very well tolerated. They get good results. Other people find it doesn't do a whole lot. It's kind of funny. It's almost black and white. With the others, it's kind of, you see sort of dose-dependent effects where the more you give, the better it works. And with pitolisant, I guess I would say maybe half the people I give it to don't really think it helps much, but the other half like it quite a bit. When we're treating cataplexy, we often treat it with antidepressants, not because we're trying to treat depression, but it happens to be conveniently that antidepressants work great at suppressing REM sleep-related phenomena like cataplexy. We think cataplexy is basically the paralysis of REM sleep that pops up in the middle of wakefulness. And so drugs like venlafaxine, fluoxetine, and clomipramine are really good. You can get typically 80% reductions in cataplexy. And we give them at these little baby doses that, for the most part, don't produce a lot of side effects. Oxybates are also really effective in controlling cataplexy, and they come with all the same caveats that I said on the last slide. And pitolisant, also good at suppressing cataplexy. In general, cataplexy is actually usually quite controllable compared to sleepiness, where for the most part we make some improvements, but rarely does somebody come back and say they're 100% alert. That happens occasionally, but it's not often the case. That's why there's a whole bunch of drugs that are now in development. And so this is a list of, honestly, this is kind of like a really golden time in clinical research for drug development in this space, because over the last 20 years, rarely was there more than one drug being studied by a pharmaceutical company, because people always thought, oh, these are just little bitty diseases. They don't really matter a whole lot. And then people are like, you know what? We got some real opportunities to make a difference here. And so Takeda, Alkermes, Centessa are all making orexin agonists. So these are drugs that bind to the orexin receptor and activate it. And what they're doing is they're binding to the orexin receptor 2, which seems to be the more important one of the, there's OX1 and there's OX2, and the OX2 seems to be more important. So that's the current main target. On the right-hand side, what you can see is what's called the development phase. And so phase 1 is the initial testing where, for instance, you'll give the drug to just healthy people who are sleep deprived and show, hey, this wakes them up. Or maybe you'll try it in a few narcolepsy patients. Phase 2 is where you'll do sort of a small to medium-sized study of maybe 20 or 30 people with narcolepsy or IH and see if you can get a good effect. Phase 3 is the big hurdle where you'll study hundreds of subjects with that particular disorder. And you got to show that the drug is not only effective in improving sleepiness, but it's also safe. And by doing a large number of subjects like that, one can then sort of launch it out into the marketplace feeling pretty comfortable that it's going to do okay. And I'm glad that, Claire mentioned that the Hypersomnia Foundation website has a listing of studies that are underway right now. And I would really encourage any of you who want to sort of help out the community more broadly to think about if this is something you can slot into your life, because this is the only way we get new meds. And as I said, most people with IH and narcolepsy are doing better on meds, but rarely are they doing great. And the hope is with some of these new meds, we're going to see substantially greater improvements. Further down the list, reboxetine is a drug that increases norepinephrine levels in the brain, and they've shown some nice clinical data in phase 2 that it really does suppress cataplexy, and they're moving on into phase 3. Clarithromycin I mentioned earlier. This is the sort of, I'll call it project from Emory University. This is the drug that's trying to reduce signaling through the GABA-A system. And I'm hoping to see more with this, but the trouble is academic places just don't have the money to do the big studies that are needed. Drug development costs tens to hundreds of millions of dollars, and that's really a hard thing for an academic group to move ahead. But my hope is that maybe they can team up with some company and make this work. Suvin is making another H3 inverse agonist like pitolisant. I can't really tell you just yet if it's going to be all that different. There's a couple at the bottom that are actually very exploratory. Where one acts upon this thing called the trace amine receptor, another activates the nociceptum system. You'll notice we haven't talked about those neurochemicals at all, and it remains to be seen sort of if those will ever actually move into humans, because those are mainly in the preclinical phase. But I have them on the list because they're really unique, and it's kind of cool to think we may have some completely different tools. Okay, so how do these drugs actually work? On the last slides, I sort of mentioned briefly some of their mechanisms, but let me just show you at this point. So there's a variety of different pathways in the brain that promote wakefulness, and we can broadly break these down into ones that make what are called monoamine neurotransmitters. And monoamines are neurochemicals like dopamine, serotonin, norepinephrine, histamine, and those are shown in the light green. And so like the tuberomammillary nucleus is the only place in the brain that makes histamine. The ventral tegmental area is a key wake-promoting group that makes dopamine. The dorsal and median raphe nuclei down in the brainstem make 5-HT, which is an abbreviation for serotonin. And then the locus coeruleus makes norepinephrine. And so drugs that increase the levels of those neurotransmitters, like histamine, for instance, can help wake you up. There's also cell groups in the brain that make another type of neurotransmitter called acetylcholine. It's also another little small molecule. And all of these systems, whether it's the acetylcholine neurons in the dark green in the basal forebrain or the light green monoamine ones, they all project pretty much diffusely through the whole brain. They go to the cortex, they turn on those cells, and when those cells are active, they turn on, ultimately, the cortex to help wake you up. Now, in non-REM sleep, which is the sort of unconscious, non-dreaming type of sleep, what's happening there is there's cell groups that are turning off those arousal regions. And the most important one is the ventrolateral, see if I get a laser here. I don't know if you guys can see this, the ventrolateral preoptic nucleus, which sits up at the front end of the hypothalamus. And this uses GABA to turn off all those arousal regions. And so when those ventrolateral preoptic neurons fire, they turn off the arousal regions and sort of help coordinate a suppression of arousal signals and help you get to sleep. Those cells use GABA, and we've talked about GABA before. GABA is sedating. Benzodiazepines, for instance, increase GABA signaling. So we can put these complicated circuit diagrams into a pretty simple model where on one hand, you've got chemicals in the brain like dopamine, norepinephrine, serotonin, histamine, acetylcholine that all help drive wakefulness. And then you've got GABA, which helps put you to sleep. And that's made in the ventrolateral preoptic area. It turns out that these systems actually inhibit each other. I mentioned in the last slide that GABA in, where's the laser? Oh, there it is. That GABA in the ventrolateral preoptic area, oh, sorry, I didn't mean to go that way. GABA in the preoptic area turns off the arousal regions, but it turns out that dopamine, norepinephrine, serotonin, histamine all turn off the sleep cells too. And so what you got is this neat little mutually inhibitory circuit where you have wake systems that turn off the sleep systems and the sleep systems do the opposite. And what that means is that when you're awake, the sleep systems are being inhibited. And that allows you to be fully awake. Whereas when you're asleep, it's just the opposite. And so these drugs actually tuck into this system. And I'll show you on a case-by-case basis how they actually work. So this is a typical nerve terminal that is releasing dopamine. And the reason I focus on dopamine is because it turns out to be one of the more important wake-promoting neurotransmitters. And so for those of you who maybe didn't have, biology in college, sorry, I don't know, I don't have a way of projecting on both screens, but of using the laser on both. But anyway, this is a nerve terminal of a dopamine neuron, okay? So there's a cell body some distance away. Nerves, they have axons that go out, they connect to a different brain region, and then a nerve terminal has these little vesicles. They're called little circular spherical blebs that contain dopamine. When this neuron is active, those little vesicles merge with the end of the nerve terminal, and their contents, the dopamine, gets dumped out into the synapse between the cells, and then the dopamine binds to cells' other neurons, and in this case, it activates neurons to help wake you up. Now, what typically happens with dopamine is that it gets released into the synapse, and then very quickly, within seconds, it gets transported back into the nerve that made it by the dopamine reuptake transporter. And so this recycling, where it gets released, vacuumed up, and then put back into vesicles again, is very typical. What amphetamines do is they block that reuptake. That's the primary thing. And so when if you're taking something like methylphenidate or Adderall or something like that, that drug is messing up the transporter in a way that it can't vacuum up that dopamine. So what happens is you have more dopamine sitting out here, and so it's a stronger dopamine effect. Amphetamines also do something else, which is kind of funny, which is they actually cause release of dopamine by messing up this movement into the little vesicles. And so there's so much dopamine floating around in the cell body that it literally leaks backwards out through the transporter, and you get a dumping, an efflux of dopamine into the synapse. And so that's another way that amphetamines can rouse people. It's also probably the source of why amphetamines are addictive, because if you take too much amphetamine, there's such a big dump of dopamine, it feels great, and it's this huge rush, which is very addictive. And that's why we don't like to use short-acting drugs so much, but rather longer-acting ones when possible. With very high doses, amphetamines can also interfere with the monoamine oxidase enzyme inside the cell, which normally breaks down dopamine. So they block the breakdown of this stuff, and so you have more dopamine floating around in here. Modafinil is actually way easier. Modafinil just simply blocks the reuptake of dopamine, so you have more at the synapse. And the evidence for this is super clear. If you make mutant mice that lack the dopamine reuptake transporter and then give them modafinil, nothing happens. It's really quite clear that that transporter is crucial for modafinil and armodafinil's effects. If you don't know this, by the way, armodafinil and modafinil are basically the same drug. Modafinil is a mixture of two molecules that are mirror images. They call them the R and the S enantiomer, but the R is the active one, and so armodafinil is the active form of the drug. The S enantiomer doesn't do anything anyway. So when you take armodafinil, you're basically taking the active ingredient of the mixed that is modafinil itself. Pitolisant is this new medicine, which is a little bit complicated. So here we have another nerve terminal, but this time it's a histamine nerve terminal. And it looks a lot like the last picture that I showed you, where you have histamine sitting around in the synapse binding to histamine receptors on the downstream cell. It's packaged in vesicles. But one important distinction, remember how the dopamine got sucked back into the cells by the dopamine transporter? There is no transporter for histamine. Once histamine is released, it just stays out in the space between cells until it's broken down by this enzyme called histamine N-methyltransferase. But it's not recycled back into the cells in the same fashion. The histamine nerve terminals contain a receptor called the H3 receptor. And this receptor normally inhibits these nerve terminals so that if histamine levels get really high, they act on this and it shuts them down a little bit. So it's kind of like a governor on a go-kart that keeps it from going too fast. And so high histamine tone acts on the histamine 3 receptor to shut down the terminals and settle down the histamine release a little bit. What pitolisant does is it blocks that feedback mechanism. So it's taken the brakes off, so to speak. So now these cells can release more histamine, but they're not going to shut themselves down in this fashion. So pitolisant enables higher histamine levels. However, so the simple story, if somebody says, oh, how does pitolisant work? The answer is, oh, it enables more, it gives you higher histamine tone. But it turns out that the H3 receptor is also found on other monoamine neurons, norepinephrine, dopamine, serotonin, and also on the acetylcholine neurons. So when you give pitolisant to varying degrees, you're getting an increase in histamine, but you're also getting an increase in norepinephrine and serotonin and other things like that too. and sort of which they are and what ratios is a source of research right now. All right, so oxybates. These are the drugs like Xyrem, Xywav, Lumryz, and the generic form too. And so, typically these are called sodium oxybate in one form or another, though there's the low sodium version. And what I'm showing on this slide here is GABA. Again, remember this little simple inhibitory molecule that's used to promote sleep. It binds to the GABA A and the GABA B receptors. Oxybates, sodium oxybate or Lumryz or whatever it is you're taking, is actually gamma hydroxybutyrate. Structurally, it's actually super similar to GABA. It's just a little bit different on one end. Instead of having an amine group on this side, it has a hydroxyl group. And so gamma hydroxybutyrate binds to the GABA B receptor. That's probably the main way that it works. It's mimicking the effects of. There's also a rumor that there may be a unique receptor for GHB, but it's kind of controversial, and people have not really figured that out. And I think it's safe to say that the GABA B receptor is probably the main source of the sedating effects. Okay, so we've covered a lot of science here, and I appreciate that this is a little bit daunting at times, but I wanted to sort of put it into this framework. Remember how we had this nice little circuit where you have the wake-promoting systems that make dopamine, norepinephrine, serotonin, and so forth? Those drive wakefulness. You have the sleep systems that use GABA. And so the oxybates enhance this GABA signaling. We don't know exactly where in the brain they work. They probably, in fact, work very diffusely. But the other wake-promoting medicines, the amphetamines, the modafinil, solriamfetol, and so forth, are enhancing the activity of these neurotransmitters. They're giving you more dopamine in the brain. They're increasing norepinephrine levels or histamine levels. And in doing so, they help directly drive wakefulness, and they're also helping enforce this suppression of sleep so you don't just transition into sleep in the middle of the day. Okay, so I'll just wrap up on the main things that we've touched on. This is supposed to be about the science of hypersomnias, and I think, as much as I hate to admit it, the science is limited to narcolepsy type 1. We have a lot of speculations about the other things, but in narcolepsy type 1, it's very clear that it's caused by a loss of the orexin neurons. The cause of narcolepsy type 2 remains unknown. It might be due to a partial loss of these cells, but we need to study more brains from people who die of other causes to actually see if these cells are really lost. Idiopathic hypersomnia could be due to a variety of things. I gave you 4 different hypotheses. One is enhanced GABA signaling. Another is some chronic inflammation. Maybe there's a lack of circadian waking drive. I also mentioned this overlap with psychiatric stuff. I'm not saying that most people with IH have a psychiatric problem. I'm just saying that clinically, it's something we try to tease apart. And many of the meds that we use to promote wakefulness actually do it by increasing dopamine in the brain. The amphetamines, modafinil, and so forth all increase dopamine. In contrast, oxybates increase deep sleep via these GABA B receptors, and by some weird magic that we don't understand, over the course of weeks or months, people start feeling more awake. But we don't really understand that in much detail yet. So anyway, I'm going to wrap up with that, and I would be happy to take any questions at this point.